Okay, so let's regroup and talk about strategy and what you might have found out.
Saying it's the only polar, so it's mainly hydrogen.
We're working on two goals here. One is teamwork, so you're working in teams, and the other is failure. So we'll see how that goes. Arturo, give me a number, two numbers.
5, 7.
5, 7. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 6, 7. Hello.
Does that mean that? Okay. What's the question?
Oh, right there. You're not who I pointed to, but you're perfect.
Sorry.
Who's your team? Ma'am, who are your team members?
This is Michelle and Justin.
Michelle and Justin. Hello. And let's talk about how you went about trying to identify. Are you comfortable talking about how you identified?
Sure.
Yeah. So what was your strategy?
At first glance, we just only considered any peak that was above, or like approximately above 1.6.
Uh-huh. So to narrow it down, you first looked at the hydropathy plot and looked at peaks that were above 1.6. Uh-huh. And how many did you find?
We found 2.
2? Okay.
Yes.
You found 2 peaks that were above 1.6.
Yeah, well, we were considering, there was one that we weren't sure about at the 400 position, but we were like,
So 2, maybe 3.
Yes.
How many people found 2, maybe 3? How many people found 2? How many people found 3? You either found one or the other. Okay, so roughly 2 or 3. And what were their positions of the ones that you identified?
For sure, approximately 20 and 800.
20 and 800. Okay, so it looks something like this. So around 20 right here, around 800, and then you weren't sure about this one because it's sort of right there, right? So some of you may have said, okay, there's 3, that could be 1.6. Other of you said, nah, it looks too, I don't know. I would definitely look at that third one because it's somewhere in there. So great strategy. And this is an efficient way to start with the hydropathy plot. 2 more numbers.
2, 7.
2, 7. 1, 2, 1, 2, 3, 4, 5, 6, 7. Hello. Who are your team members?
Hi. This is and this Paul.
Hello. Did you also come up with these right
Yeah,
here?
we
And
did.
what was your next step?
So we were looking at the position, and then we went to the receptor sequence
Uh-huh.
and looked for, I guess, like ranges that included like 20 amino acids that were hydrophobic and hydrophilic, but not charged.
Okay.
Yeah.
So what you did was, just to repeat for the back, because it doesn't, your voice doesn't project to the back, is you took these peaks, you started with the hydropathy plot also, then took that and looked at the amino acid sequence, went down here and looked around 20 or so, around 400 and around 800, and looked for 20 or so a stretch that lacked, somehow I'm having trouble getting these words out, a stretch of 20 that lacked charged amino acids. Perfect. 2 more numbers.
1, 2.
1, 2. 1, 2.
Oh,
Hello.
hi.
Hi, Joanne. Who are your team members?
This is Azir, and this is Monica.
Hi. Is that what you did also?
Yes. So after narrowing it down in the hydropathy plot, we just tried to look at the sequences that correspond to those areas and search for like just amino acids that were.
Great. And how many did you find? Where'd you get that part?
We actually only found one.
You found one stretch
Yes.
that satisfied your rules.
Yes.
Uh-huh. And your rules were that it be above 1.6 and it be greater than 20. So which of those, did you underline the amino acid sequence?
Yes.
And what is this potential sequence?
We thought that it would start around the last part of 790, so the A and the L, and then all the way until the end of 810.
Okay. So this team looked to see what, underlined an amino acid that was around 800, found a stretch there that was around 800. The others were too short. So I think if we look at that first stretch right here, those of you, you probably underlined, those are charged right here. This must be the stretch that's giving that hydrophobic index of above 1.6. But when you looked at that, that was below 20, so you rejected that one. And then you probably looked around, maybe you were one of those that said that's close enough to 1.6, which I think it's better to be conservative, and looked there, and that one was also less than 20. So these cannot be, those cannot be transmembrane domains. And then around here, right here, you found a long stretch, right, exactly what you said, that right here, so that's a potential hydrophobic domain. So it looks like the LDL receptor then, you're faced with this, it's so powerful, this prediction. You're faced with these 860 amino acids that you're gonna scan one by one to try to find transmembrane domains. By coupled with the hydropathy plot, you can come up with a really good guess of what the transmembrane domain might be. Looks like the LDL receptor spans the membrane a single time, with a very large domain on one side of the membrane. We don't know if that's the outside of the membrane or the cytosolic side, and a very tiny other domain, either on the outside or the inside, spanning the membrane a single time. You could take this amino acid sequence right here and run it through a sequence prediction, predicting software to see if that is indeed an alpha helix. And that would help confirm whether this is a potential hydrophobic domain. But note that I'm using the word potential, because all of this is just predictions. We don't really know that this is the transmembrane domain of the LDL receptor until we do experimental tests in real life to look and analyze the transmembrane domain. But we have a really strong hypothesis right now. There's a single transmembrane domain and a very large domain on one side and a smaller domain on the other side. Any questions? Yeah?
So the threshold is 20?
The threshold for this course is 20. It has to be 20 amino acids in a row that do not have a charged amino acid in there. And altogether, those 20 have to correspond to a hydropathy plot above 1.6. And you'll be playing with hydropathy plots in homework that you'll be doing, I think this week probably, right here. And you'll not only, we've talked about the rolling window, you can play with that window and see what happens if you expand the window. Obviously, if you expanded it to 800, that's going to be ridiculous. But you could expand it to 3 or 5 or 10 or 20 and just play with it to see what happens. That's what scientists do in real life. Okay, questions? Y'all did really well. Good teamwork. We still haven't had failure, so that's too bad. We will. Okay, so as I said, these are all predictions, and in order to really study the biology of receptors, we need to actually do experimental data. And the experimental data we do is largely biochemistry. The LDL receptor was isolated is in high abundance in liver, and Brown and Goldstein, Joe Goldstein and Michael Brown at UT Southwestern pioneered the work on the LDL receptor as well as cholesterol metabolism. It's a fascinating story. Again, we're going to be revisiting that story throughout the course. And their work, they ended up winning the Nobel Prize for their work on the LDL receptor and cholesterol. And the way that one tool that they used was biochemistry. So what they did was exactly what we've talked about with cell fractionation. You can take liver cells, mush them up with a pestle, and then spin them down, do various spins, a low-speed spin and a high-speed spin, to isolate membranes. So the membrane, the plasma membrane, where the LDL receptor is, would be in the very highest speed pellet. And then you can begin to analyze whether these proteins are on the domains, the respective domains, where they are. Are they on the outside of the cell, or are they on the inside of the cell? So in order to do this sort of biochemistry, we have to treat cells with different reagents that will help us extract the proteins so that we can look at them intact. And also, these various treatments allow us to treat cells to figure out whether proteins are integral membrane proteins, peripheral membrane proteins, what domains are on the outside, what domains are on the inside, etc. So let's look at that experimental data. It's important to understand that integral membrane proteins can be isolated from membranes with detergent. And we've talked about a very strong detergent, SDS, right here, which structure is shown right here. Look at that large hydrophobic tail right here, and then it's charged. And you know that SDS actually is such a strong detergent that it denatures the protein. So it completely unwinds the protein so that it is just a single snake covered with SDS. In order to study membrane proteins and retain their character, we need a more gentle detergent. And two most commonly used gentle detergents that are used to study membrane proteins are Triton X100 and beta-octylglucoside. We're going to mainly talk about Triton X100. If you compare the structure of this gentle detergent, Triton X100, with the strong detergent, SDS, you see that it's a little bit less, it has a shorter hydrophobic portion, and it has a polar head group that is not charged. It's polar because of that little OH group right there, right here, whereas SDS is charged. Whether we're talking about a strong detergent or a gentle detergent, they all are not miscible in water because of these hydrophobic tails. And so they form these structures called micelles right here, where if you put this detergent in water, they'll sort of circle around each other to shield that hydrophobic tail away from the water. It's just a natural physical thing that detergents do. And what you see right here is here in pink, in blue is water. You can see the hydrophilic head groups right here, which would be this part of SDS, or this portion of Triton X100. And they've put their tails sort of in the middle right here to make a hydrophobic core right here. This is called a detergent micelle. Water on the outside, fatty acid stuff in the middle, sort of like an M&M, candy on the shell on the outside and the chocolate on the inside, shielded away from your mouth. Or hands, I guess it is. No, it's in your mouth, not in your hands. Okay, so that's a detergent micelle. And this structure, doesn't it look a lot like a phospholipid bilayer? And that's why these detergents are so great at extracting membrane proteins, because this is what they do. If we take a gentle detergent, and I've added these words right here, and you might add it to your slide right here, gentle detergent, so like Triton X100, and you look at the bilayer right here. So in this diagram, what we have is a bilayer with the membrane protein in it. This has been extracted from, this has been prepared from a cell through centrifugation, the cell fractionation we talked about. So we have this membrane pellet, and we add to it detergent. This shows the detergent right here. This is in buffer, right? This whole thing is in buffer. We're adding them together. We see the detergent micelles, which have their polar head groups outside shielding this. But it's an equilibrium, so these are coming in and out, largely in this state right here. But they come in and out with some equilibrium. The equilibrium is forced right here because of that hydrophobic moiety. If we mix these two together, what happens is with some frequencies, detergent monomers actually start integrating in with the membrane protein and start interacting. These, the hydrophobic tails of the gentle detergent